Hi everyone, thanks for listening to my presentations. Today I'm talking about liver functions and liver enzymes. That will take me through the functions, the liver function test, and why the liver function test is different from liver enzymes. Remember, I have published a brief anatomy of the liver and also the biliary system at a glance. Today is all about functions of the liver and why liver function test is different from liver enzymes. Okay, let's go. One prominent function of the liver is the amination of amino acids. The amination takes place in the liver to break down amino acids for energy. The amino group is further converted to ammonia, leaving carbon and hydrogen behind. That could either be recycled or oxidized for energy. The produced ammonia is converted to urea and uric acid when carbon dioxide molecules are added. The urea and uric acid can safely diffuse into the blood circulatory system and excreted in the urine. Synthetic capability of liver involve plasma protein synthesis, example the albumin. All clotting factors are produced in the liver except two, which are factors 8 and von Willebrand factor. As far as excretion is concerned, liver can excrete the bilirubin, cholesterol, hormones, and drugs. Part of the functions of the liver is enzymes activation. Enzymes activation. The storage capability of liver involves glycogen storage, vitamins, and minerals. When it comes to metabolism, virtually anything that goes through the mouth will pass through the liver. Metabolism of fats, proteins, and carbohydrates, including the medications or drugs that we take. And that will take us through what is known as first pass effect or pre-systemic metabolism. When you take any medication by oral, the metabolism will go through the liver. Another function of the liver is bioproduction and excretion. I'll not waste your time here repeating the same thing. You can just check my channel for the biliary system at a glance, and you will understand fully. Another function of the liver is blood detoxification. Toxins are dangerous to normal functioning of all the systems in the body, and the liver helps greatly in detoxifying all toxins in the blood. Okay, now liver function test and liver enzymes. Why are they different? And what are the differences? When it comes to liver function test, the doctor is trying to find out if this liver is still capable of functioning properly. Okay? Then we'll be calling for the value of PT or INR. It's the same. It's either you call for INR or for PT. Then APTT. When we check for these two, we are trying to find out what is happening to clotting factors. Remember, I said earlier that the liver is responsible for the production of all clotting factors except two, that is von Willebrand factor M factor 8. So we want to know if the liver is still capable of producing those clotting factors. If not, we'll be dealing with massive bleeding. We also want to know if the liver is capable of albumin production. And of course, what is happening with bilirubin? Okay, particularly conjugated bilirubin. Then how about the total protein and glucose/glycogen storage capability? Or we are even dealing with glycogen storage disease. All these are pointing to the functional capability of the liver. On the other hand, when the question is about liver enzymes, then we'll be dealing with AST, ALT, GGT, ALP. But without even going into details of each of them, liver enzymes will only give clue as per the degree of injury when the liver function capability could still be preserved in many instances. Let me explain. Liver function test will give clue that this liver is badly damaged, cannot function properly. Okay? Then you have what is called hepatic impairment or liver failure. But in liver enzymes, the value will give us clue that there's an injury, there's an insult to the liver. Maybe it is hepatitis. But the ability to function is still preserved. However, when we have very, very, very high values of liver enzymes, that should lead us to find out what the liver function is right now. So the first thing we should do is get liver enzymes. When they are raised and pointing to severe liver injury, then we should call for liver function test. So when the result is out and the liver function test is showing, oh man, this is bad, then we know we have trouble at hand. Because the liver enzymes will give clue as per the degree of injury that may have a preserved liver function. Even it is not all liver enzymes that are pointing to trouble in the liver, because some, like ALP, that's alkaline phosphatase, or GGT, gamma glutamine transferase, are non-specific to the liver. In other words, they could be found in some other parts of the body under different situations outside liver injury. Like ALP could be problem with the bone somewhere. So it is not specific to the liver. GGT is not specific, but if you find it raised and the problem is pointed to the liver, it could be cholestasis in ALP, and GGT could be pointing to both cholestasis, viral hepatitis, and if it is about 10 times raised above the normal value, it could be pointing to alcoholism. So, in summary, excessively raised GGT could mean we are dealing with alcoholism here. And ALP is non-specific, but when we find it, we could be thinking of cholestasis somewhere. And of course, AST and ALT, we could find the ratio of AST and ALT also to give us clue if we are dealing with viral or we are dealing with alcohol-related injury to the liver. Then we'll call for liver function test to know how badly damaged the liver is to the point of not being able to function, or the functional capability is still preserved or not. And with that, I've come to the end of this short presentation. Please remember to share, remember to subscribe. I have started the journey into the largest organ in the body called the liver, and I will continue with a series of presentation on liver-related pathology. Thanks, and I appreciate it.